good.
Good evening, and welcome to tonight's lecture. I'm George Kraw. Joining me as moderator tonight is Professor Susan Carter, the Shinsheimer Chair of Molecular Biology. Susan studies innate immunity and inflammation. Her work is mapping how immune cells develop and respond during interaction, infection claims to identify novel drug targets to contribute infections, to combat infections and inflammatory diseases. Welcome, Susan Carter. Carter.
Thanks so much, George. Yeah, it's my absolute pleasure to get to introduce you all to Professor Carol Greider tonight. So Carol really is like an incredible role model for the UC system. That's where she began, and it's where she has made her way back to. So Carol started off her career at UC Santa Barbara, where she got her degree in biology. And then she went on to do her PhD in molecular biology at Berkeley, which was her seminal work that she continues to this date. And from there, she moved to Cold Spring Harbor in 1988 to start her independent career. And she really quickly rose up through the ranks there and then moved in 1997 to Johns Hopkins, where she, again, quickly moved through the ranks and became chair there, where she was chair for 21 years, was it? Yeah. Yeah, before she moved back to the UC system, and we're so all incredibly lucky that she moved back to UCSC in 2020. So Carol has won an incredible number of honors and awards. And so just to kind of highlight a few, she was a Pew Scholar. In 2003, she became a member of the Academy of Sciences. In 2006, she won the Wiley Prize in Biomedical Sciences, as well as the Albert Lasker Award for Basic Medical Research. And then in 2009, she won the Nobel Prize in Physiology or Medicine. And it's really incredible to think that she's one of 13 women to have won that particular award. So it's a special group. So I could go on about the awards all night, but Carol is so much more than the awards. I really think of Carol as a scientist of the people for the people. She's one of the most modest people I have had the opportunity to interact with, and she really does utilize her position to promote others. She's an amazing, staunch advocate for women in science and for minorities in science. And at UCSC, she, is really committed to this work. She's part of our Genomics Institute Diversity, Equity and Inclusion Committee, our DEI, as well as our department's DEI committee. And there really is no job too small that she won't get involved with. She's one of the first to offer to get the pizza or pick up the coffee. And I think that's really important. Our students see that this superstar is willing to get her hands dirty. And so I think when I interact with Carol, I would say she's equal parts super fun as well as intense. She's firm, but she can command a room with the lightest touch. I seen that recently. We were having a meeting that I was a bit worried was going to go off the rails, and Carol just commanded the room in about a couple of sentences. She got everybody back on track, and it was an amazing meeting. Afterwards, I joked with her that I was like, I think you might now be in the running for a Nobel Peace Prize. To your Nobel Prize. So maybe in her spare time, she can work on world peace. I promised Carol I would keep it short. We're so incredibly lucky to have her at UCSC, and you're all in for a treat tonight to get to hear Carol. So without further ado, Professor Greider.
Thanks, Susan. Is the mic on? Great. Thank you. That was a really wonderful introduction. I appreciate being here to talk to local colleagues. It's always harder to talk to local colleagues than it is to talk to people that you don't know. And so I just want to make one small disclaimer, and that is I'm trying out this new technology here so that the people on Zoom can see the pointer just like the people in the room can. So bear with me. I'm going to try this out, and we'll see how it goes. So I wanted to give a chance to tell you some of the background about telomeres and telomerase at a very general level, and then take you through, in the last 10 or so minutes, some of the really interesting new things that we've been finding in telomeres. So we'll start with chromosomes. So let's see the pointer. All right, this is the technology I was telling you about. It's gone now. This is a cell. Within the cell, there's a nucleus. And within the nucleus, there are chromosomes. The chromosomes are made of DNA. I'm not going to be able to do this, I think. I'm not going to be able to pull it off. And the DNA encodes for all of the genes that allow the cell to function. So the T, C, A, and G of the DNA specify the code for the molecules that carry out function in the genome. So we will be talking about DNA sequences, but we're not going to be talking about the genes for the DNA sequences. Instead, today, we're going to be talking about the ends of chromosomes. So telomeres are the ends of chromosomes. So all along the length of this chromosome, there are genes that encode the various proteins. But at the very end of the chromosome, I think I might have to punt to one of those actual pointers. If somebody, do you have an actual pointer? Because the people in the room will be able to see it at least. I was going to try this, but the lag is pretty long. So the ends of the chromosomes are these telomeres here, shown at the end. And they are made of DNA, but they don't code for anything. Thank you very much. OK. So sorry, all you Zoom people. You won't be able to see what I'm pointing to, but the people in the room will be able to see. So we're talking about the telomeres, which are the ends of chromosomes. And they play two important roles. They have to protect the chromosome ends, and they have to maintain telomere length. All right. They have to maintain telomere length. So there are DNA sequences on the ends of the chromosomes. And the human telomere sequence consists of a tandem repeat of a very simple sequence, repeated over and over and over. And that sequence is TTA-GGG. I'm wondering if there's one that has a battery that's working. There we go. TTA-GGG, over and over. That's what's at the end of the chromosome. It does not code for anything. It's not like it encodes a particular protein, but it plays an important functional role at the chromosome end. So telomere repeated DNA units are lost at each cell division. So this is the chromosome, all of the DNA sequences along the length of the chromosome. And these are those repeated TTA-GGG repeats. With each round of cell division, there's a little bit of sequence of these repeats that are lost. And this has to do with the mechanism of how the chromosome is normally copied. So the mechanism can't copy out to the very end of the chromosome. And so as a consequence, these sequences are lost. This enzyme that we call telomerase will rescue that problem. Because if we're all sitting here in this room and your chromosomes are getting shorter and shorter and shorter, there would be a problem. So we need to figure out how to solve that problem. So you have this replication that leads to telomere shortening. And then you have this enzyme that we call telomerase, which adds repeats back on to the ends of the chromosome. So that the short telomere now can be elongated because this enzyme is adding those TTA-GGG repeats onto the ends of the chromosome. The telomerase enzyme is this beautiful structure here, which is made of both RNA and protein. So there's an essential RNA component of this enzyme, as well as an essential protein component. And so this gives me now my bona fides to be part of the RNA center at UCSC. So this was my connection, because this enzyme telomerase actually is composed of both RNA and protein, and it adds these telomeric repeats onto the ends of the chromosomes. So if we look at a whole bunch of chromosomes, again, here is the coding region of the chromosome, and then each chromosome in the cell has a different number of these telomeric repeats, because there's some shortening due to replication, and then some lengthening, and some shortening and some lengthening. So there has to be an equilibrium level of the telomere length. So if you look at all the chromosomes in a cell, they're going to have slightly different numbers of repeats. And the way that we look at this historically has been on this southern blot. So here is a size marker at 3 kb, and here's a size marker at 1 kb. And you can see that the telomeres themselves have a distribution of lengths, just like we see here, that there are some telomeres that are shorter and some that are longer. And so the way that we look at this is that here is the length of the telomere, and here is the frequency, or the number of ends that have that length. So there are a few telomeres that are very short, a lot of telomeres that are medium length, and a few telomeres that are very long. So there's this distribution because of the way that the telomeres are shortened and then lengthened and then shortened and then lengthened. Okay, so we're going to have a couple of notes along the way in this talk. The first note is that telomere length measurement method number 1. We're going to be talking about different ways that people measure telomere length, and that's the southern blot. So here's an example of the southern blot of telomere length with this distribution of lengths for yeast, and we have this nice frequency distribution here. Human cells also have a distribution of telomere lengths. Although they're much longer, you still have this heterogeneous smear of the telomere length when you look at human DNA. So that's conserved across various organisms. So this is foreshadowing here. So the human telomere length on a southern blot. So that's what I'm showing you here, and we'll be getting more into methods that we look at human telomere length on. Why does telomerase matter? Because it's required for all cells that must divide many times. So there are two crucial areas here. One has to do with normal tissue renewal. So you think of blood cells, or you think of skin cells, where you have some cells that die, and then others have to renew that tissue. So we have, we have a stem cell here, which has to make more copies of itself so it can renew the tissue. And then there are cells that come off of those cells, which are the progeny. And in order for those cells to divide that many times, they have to solve this telomere problem. If they don't solve the telomere problem, then the telomeres are going to get to be too short. The other critical area where telomerase matters is in cancer. Again, in a particular tissue, you will have a stem cell that can self-renew, and then you have various differentiation products of those stem cells. And if a particular cell gets a mutation that causes it to divide more times than it normally would, for those cells to go on and divide as tumor cells, they have to solve the telomere problem. So telomerase plays an essential role both in normal tissue renewal as well as in cancer cell growth. I'm going to be talking tonight a lot about the normal tissue renewal, and we can talk in the questions and answers about cancer. I'll touch on that a little bit, but I won't go through as much of the evidence, just to keep things succinct. So in order to ask the question, what happens when you don't have telomerase, to ask the question about what is it necessary for, we generated a knockout mouse to study telomere function. So we created a mouse where we got rid of the telomerase entirely. And what's shown here, again, is a foreshadowing about how we looked at these telomeres. These are the mouse chromosomes. The actual chromosomes are shown here, and the telomeres are highlighted in red. So that's how we studied this mouse that doesn't have any telomerase. So we took the mouse that was the first generation, the G1, that has no telomerase, and we crossed it to another mouse that has no telomerase, 2 G1s, and then from that genetic cross, we get the G2 mouse. And you can see here, again, this is the chromosome, and these are the telomere repeats on the chromosome. There's a certain length of these telomeres in the G2 mice. We then take those G2 mice, cross them to each other to get the G3 mice. And what we find is that the telomeres are now shorter when you don't have telomerase. We take these G3 mice, cross them to each other, and get G4 mice. And we find that those mice have telomeres that are shorter again. And so this can only go to the G6, and then we can't get any mice anymore. So this shows that actually, when you get rid of telomerase, you really do cause this shortening that I was talking about that normally compensates for the telomere length. So in these short telomere mice, the short telomeres cause a loss of tissue renewal capacity. So in the blood, we find that there's bone marrow failure. In the intestine, which is another cell type which has to divide to have continual renewal, we see loss of the cell lining. In the skin, we see decreased wound healing. In the hair, we see premature graying. And in the testes, we found cell death that caused infertility, and thus we couldn't continue the mice any longer. So this is what the telomerase null mouse showed us, is that when you have the chromosome with the telomeres, as the cells are dividing and those chromosomes are replicating without the ability to elongate them again, they get to be short, and when they get to be very short, they're no longer functional. And what happens is that those cells undergo cell death or another form of cell death called cellular senescence, just 2 different molecular ways of having the cells stop dividing. So the important thing here is that there's a buffer zone. There's nothing that happens until the telomeres get to be short. So it's a threshold. When the telomeres reach a very short threshold, then the cells die. So here's another method. Note, telomere length measurement method number 2, quantitative FISH. I showed you this image before with the mice. So here we take mouse blood cells and we display the actual chromosomes. And you can see that the red on the ends of the telomeres represent the amount of telomeric repeat on the ends of the chromosomes. So you can actually quantify the intensity of this signal and the intensity of this signal, and individually look at all of the intensities of all of the signals. And that is the method that we initially were using to look at the telomere shortening in these mice. So by looking at all of these intensities, you can look at the signal intensity versus the number of chromosome ends that have that intensity, and you see the same distribution that I was showing. So this is one method that was developed by Peter Lansdorp in Vancouver, Canada, to measure the telomere length by looking in the microscope. A number of years ago, we were interested in what happens in human cells when you look at the telomere length in white blood cells in human cells with age. So this is an example of the median of that telomere length. And here is the age of the individuals from which those blood cells were taken. And we can see that there's a progressive decline in telomere length in these white blood cells with age. At first, we thought that there may be no telomerase there, but now we know that there's just not quite enough telomerase. So the number of cell divisions is more than you can keep up with the amount of telomerase that's adding onto the ends of the chromosomes. So we'd been studying these telomerase null mice to understand what is the function of telomerase. And I was at Johns Hopkins University, And a colleague of mine called me up and said that there was a patient that came into the clinic that had this disease called aplastic anemia, which is bone marrow failure. And this is the patient that came into the clinic here. And my colleague, Mary Armanios, was an MD in the lab, and she got very interested in this family. And she was able to identify this individual who had this bone marrow failure syndrome, and then enroll many of his family members to understand the genetics of this disease. So here's the grandmother. The circles are females, the squares are males. Sorry, that's just how it is. So here's the grandmother. The ones that are colored in black, it turns out all of these had a mutation in telomerase. So they were deficient for telomerase. Not null, but deficient. And so you can see here's the grandmother, the son, the grandson. And the very interesting things about these families is that there was progressively worse disease with the later generations and progressively shorter telomeres with the later generations, which is what we had seen in the telomerase null mouse. So this is a case where we made the mouse to study a very fundamental question, and then we found out there was a human disease that was associated with it. Normally, you would have a human disease, and you would go and make a mouse. But we had already pre-made that mouse, so we knew all about the biology of what would happen if you don't have telomerase. And it mapped very well. The human disease mapped on what we'd already found in the mice. So Mary now got very interested in these families. So some of the mutations are in telomerase. Some are also in other components that are necessary for telomerase action. So we call those other telomere maintenance genes. She now has 140 families that have this inheritance pattern. And so she can look very carefully to understand what happens when you don't have enough telomerase. And what she's found is that there's this bone marrow failure, which is what this initial patient had shown. She also found that pulmonary fibrosis is actually the most common thing that occurs when you don't have enough telomerase. Immunosenescence, so an insufficient immune response. Emphysema susceptibility, something called cryptogenic liver cirrhosis, and gastrointestinal disease. And so all of these are commonly found in these families that don't have enough telomerase. And the interesting thing is that there's this decreased telomere length and increased severity of disease with each generation. So here's the next note. Telomere length measurement method number 3. So what's shown here is a cell nucleus. The blue is DNA. And each one of these dots here within this blood cell is the same thing as those dots on the ends of the chromosomes I showed you. It's the same method. You can look at how much telomere repeat is there, but now you're doing it for the whole cell. So you can quantify how much telomere is present in one cell versus another cell. And this method, called FlowFish, determines the total telomere repeat content in blood cells. This is now the standard method for clinical-based analysis. If a family comes in and you suspect they may have short telomeres, FlowFish is what you would do to determine clinically. And Mary established a clinic at Johns Hopkins, and also Peter Lansdorp has a company, Repeat Diagnostics, that will do this. And those are the two clinically approved assays in the country. So what did Mary find? So if you look at individuals of different age, and you look at telomere length with this FlowFish assay, you can look at here at 0 age. This is cord blood, so people when they're first born. And here are the different ages of individuals. So you see, as we had seen back 25 years ago, that there's a shortening of telomere length with age using now this FlowFish assay. And to be able to clinically diagnose disease, you need to put confidence intervals on these data. And so you can see the 99th percentile in the population for long telomeres, the first percentile for short telomeres. Most of the individuals are around the 50th percentile. So this is what we currently use to diagnose disease. And this comes from, again, blood cells taken from these individuals and mapped onto the telomere length. One of the things that you should note is that within a particular age group, there's a normal variation in the population. There's not a unique length, just like there's a normal variation of height. If we looked at how lined up all the normal people in this room, are there normal people in the room? Okay. If we lined up all the people in the room, you would see various different heights. That this is normal variation that occurs. It's not pathology. So this is a key to what some of you might be wanting to ask me later on about short telomeres and telomere shortening. That it's not that there is a particular telomere length that you should be at a particular age. There's a distribution in the population. So from Mary's work, she showed that people with defects in telomere genes have shorter telomeres and are at risk for age-related diseases. So here are some families from her work. The mutation carriers are here in the very low percentiles, below the 10th percentile. And their family members that are not mutation carriers are around the 50th percentile. So again, she now has many more families and is studying these disease aspects. So telomere length equilibrium plays a critical role in human disease. Hope I've convinced you of that. There's a short telomere threshold where you have stem cell failure. So in this distribution of lengths, if you fall below this length, then you have this cell death, which leads to the inability of self-renewal. On the other hand, long telomere threshold predisposes to cancer because, again, those cells can divide more times than they normally would have, and that contributes to cancer. So disease can result from telomeres that are too short or too long. So what we wanted to know is, how is telomere length equilibrium established? If you don't understand the mechanism of how it occurs, you can't intervene in disease. And so we wanted to go back to fundamentals to understand how does it actually work, and then maybe we can make changes. So there's very good evidence that all telomeres shorten at a similar rate. So you have this shortening that occurs through natural divisions. And the models that people have, for which there's some evidence, is that the shortest telomere is preferentially elongated. So you have these short telomeres that are more elongated, and you have all of these guys elongating, and that somehow establishes this equilibrium. But that's not a very mechanistic understanding. That's just description of what we see. So we wanted to understand, what is the molecular mechanism? What are the fundamentals that allow telomeres to be elongated? So we set out to try and understand these fundamental aspects of telomere length regulation. And a PhD student in my lab, Sam Scholes, who was at Johns Hopkins, was devising interesting new methods to look at these mechanisms. And what Sam started to do was to use this new technology called nanopore sequencing. And starting with nanopore sequencing, she was studying telomere length. So what is nanopore sequencing? And this is the theme of tonight. With new methods comes new insight. This new technology of nanopore sequencing allowed us to see something that we couldn't see before. So nanopore technology allows very long read sequencing. Importantly, it was invented here at UCSC. So David Deemer, who was going to be here tonight. Hi, David. Sorry you couldn't make it. He told me he was going to come. And Mark Akeson with their colleague Daniel Branton. And here's a picture of their recent book. So this is a completely new technology that they devised by which you can take DNA and you can have this little small pore, that's why it's called a nanopore, and you can sequence the DNA as it goes through the nanopore. So this technology is revolutionizing genomes and genome sequencing and understanding genome sequencing, but we wanted to use it to understand telomeres. So let me tell you just a little bit about what this does. So Nanopore long-read sequencing changes the landscape for genomics. So all of the sequences you've heard about the revolution in genome sequencing and all these sequences that have been around, that's all been done by what we call short-read sequencing. So this is about 150 base pairs. So every time you look at a piece of DNA, you get 150 base pairs, 150 base pairs. A different run will give you a different 150 base pairs. And so you can line these guys up and make these longer contigs, as they're called. And then you line all of those up to get your genome. The short-read sequences gives you these genomes with some gaps, because sometimes you don't cover the whole area. Long-read sequencing is up to 30 kb. So look at the difference, 150 base pairs, 30 kb. Pretty big difference. So you now have these long reads. These are the sequences that you're actually getting. And then you can make your contiguous regions by lining them up. And now you get genome sequencing without gaps. And so this is really changing how genomics is done. This is what most people are thinking about for nanopore sequencing. There are many other revolutionary things that nanopore can do, but this is what people have been thinking a lot about. So we were interested in, could we measure telomere length, and could we get at the mechanism by measuring more accurately the telomere length? So what Sam did was to make chromosomal DNA, and this is my little artistic rendition of chromosomes. So each one of these colors is a different chromosome. And what she did was, I'm just showing one of those chromosomes. She put a little tag onto the very ends, which are the telomeres. She did that for all of the chromosome ends. And then she could take and sequence everything with Nanopore. And you get very, very, very long reads with Nanopore. And the importance of that is that this is the telomere length now as a DNA sequence, which is highly accurate. But of course, that telomere is attached to a particular chromosome end. And so the read would go all the way through the telomere and include the specific chromosome. So now we have chromosome-specific information that we didn't have before. Oh. So we get telomere length and the chromosome identity. And then we can plot, just like we had plotted before. If you look at all of the telomeres, you get the same length distribution that I was showing you before as a distribution like this. So this is all of the telomeres. You get this distribution, which is centered here around just below 400 base pairs in yeast. And the surprise was that in 3 different cells, 3 independent cells, all of the copies of chromosome 1L were shorter than this. Whereas all of the copies of chromosome 2, so this is chromosome 2, the left telomere, chromosome 1, the left telomere, chromosome 3, the left telomere, were significantly larger than the average. This was not what we expected. When we looked at all of the different chromosomes, these 3 independent samples were reproducibly lower, and other 3 independent samples were reproducibly higher. Okay, I've been working on telomeres for 33 years. I did not expect this. We always thought that there was a uniform distribution of all of the telomere lengths. So this new technology allowed us to see these chromosome end-specific telomere lengths and their distributions, just as we had seen, but it's not an overall distribution. It's a specific distribution. When we first saw this, we thought, nah, couldn't be true. We've been studying telomeres for 33 years, and we'd never seen this before. And we'd never seen it before because we didn't have the method to see it. All of the methods I was showing you before only show the global telomere length. But still, we thought, can't be true. Let's do another experiment. So Sam took a single cell, put that into culture, and grew it for 120 doublings. So that's huge number of cell population doublings. and then took a single cell, and a single cell, and made DNA from those cultures to ask, maybe we just hadn't come to equilibrium yet. Maybe you see those differences just because it's just a random distribution. But what she found was for each of these 4 samples, the 1L was always long, the 2L always short, the 2L was always long, and there was a consistent, all of those independent replicates were the same as each other. So this gave us confidence that this is really something that's true. So what we found is that each telomere is regulated around its own unique length. Rather than this global distribution of telomere length that everybody had been studying for the past 30 years, what we find is that some chromosomes have a distribution around this length, and some chromosomes around the orange length, and some around the purple length. So the naive idea that you can simply elongate the shortest telomere doesn't work, because the shortest telomere here is much longer than the longest telomere here is longer than the shortest telomere there. So the models that we had for how telomere length regulation would occur are all out to lunch. So we have to develop new models because we have new data. So this implies that new models of length regulation need to be considered. And so that's what we're interested in doing. The next question is, are chromosome end-specific lengths conserved across organisms? I told you a lot about human disease, so of course we wanted to understand, is this new thing that we found in our model system, yeast, does it also pertain to humans? So we were able to develop a nanopore sequencing method for human telomeres. It's a much more difficult task because the human genome is 130 times larger than the yeast genome. So we had to develop a different method to do the human one versus what we'd done in yeast. So what we found is that nanopore sequencing recapitulates the broad range of telomere length on a southern blot in human blood samples. I'd shown you southern blots before, and here is the telomere length distribution for a number of different blood samples from individual people. When we then do nanopore sequencing, we can show that the mean telomere length is recapitulated by the nanopore sequencing very much. We had to peg this to the gold standard of what everybody uses, which is the southern blot. So the nanopore sequencing readout, so each one of these dots is a particular sequencing length of an individual. So the telomere length analysis by nanopore sequencing is also highly reproducible. We wanted to know this because we know that if we develop a new method, people are going to think about using this potentially in the clinic. It's not there yet because we don't have all of the clinical assays, but we want to know how reproducible this is. So here's an example of the same sample analyzed independently 14 times. So what they call the correlation of variation, the variance of what you get as a result is 1.2%. So it's highly reproducible. My student, Kiresh Kurimanin, who developed this method, was still working at Johns Hopkins, finishing up his PhD. And he was working with Aliana Groot here at UCSC. And so we took the same samples, and he analyzed them at Hopkins, and she analyzed them at UCSC. So here's an example. Whoops. You can see this particular sample, very similar in all cases. Here's the small amount of difference between these different samples. So it's reproducible in 2 different laboratories. So we were able to look at this data, which I had shown you previously, where they had used flowfish, and this is the clinical assay that is now used to diagnose telomere patients. And so we have this data of the distribution of telomere lengths in the population. And when we do that with our nanopore sequencing, we find a very similar distribution of telomere lengths. And we also looked at the cord blood, and we found that the nanopore sequencing gave a slightly tighter distribution than the flowfish did. But otherwise, they're very similar. So then we looked at chromosome-specific telomere lengths. And in fact, we did find that human chromosomes have N-specific telomere lengths, just as we had found in yeast. So the difference here is that our yeast, what we call haploid yeast, that is, they had one copy of each chromosome. Of course, humans have two copies of each chromosome. You get one from your mom and one from your dad. So if we look here, the bulk is all telomeres. So this blue here is the average of all the telomeres, which is what you would normally see in all of those other assays. And let's look here at chromosome 1, P, which is one of the arms, from the mother, the maternal copy, or chromosome 1P from the father, the paternal copy. So here is the telomere length distribution of the mother, and here is a telomere length distribution of the father. This, to me, is astounding. The average is 4.5 kb, and the difference is 6 kb. We did not expect this. We did not expect there to be allele-specific differences as well. That's not true for all chromosomes. So these are all color-coded. So the two copies of the chromosome are the same color. So here, example, is 3P, the mother and the father chromosomes, and they're the same as each other. So some chromosomes are the same, some are different, some are longer than the mean, and some are shorter than the mean. So
The 312s up there?
3 12s.
There's 3 under 12. MP and a second P, blue.
So we don't do left and right in humans. We do P for petite and Q for not petite. So this is 12P, and that's 12Q. It's very difficult because the P's and the Q's, I get my P's and Q's mixed up all the time. Mind your P's and Q's. So this is very surprising, and this is just recently sent off for publication, so this is very new. Of course, we have competitors on this because we published the yeast paper, and then other people are going to want to do the same thing in humans. And amazingly, there's another group led by Jan Karlstetter at the Salk, and also Nanopore. So Nanopore is now on this as well. And if we take this data from these exact cells, they did the same experiment, and they also posted it on bioRxiv, and it's the exact same result. So that gives some confidence. So each telomere is regulated around its own unique length. Here's the general telomere length that we always understood before, and now we understand that there are chromosome N-specific lengths. So now we can explore what this means for telomere length in human disease. If you don't understand what the actual length is, you can't hope to make changes for disease. So what I've told you is that telomere length is maintained as an equilibrium distribution. Short telomeres contribute to age-related degenerative disease, while long telomeres predispose to cancer. Previous methods to measure telomere length could not distinguish specific chromosome ends. Nanopore sequencing allowed us to detect specific chromosome N-specific telomere lengths. New mechanisms for telomere length regulation can now be discovered. With new technologies are transformational in moving past old paradigms. So science is teamwork. I didn't do any of this by myself. Here are a number of pictures over the years as we were doing all of that work in the mouse and understanding the human disease. And the most recent players here are Kiresh and Sam. Sam did all of the yeast work. They helped with each other's work. And Kiresh has the recent human data. So this is the Nanopore sequencing pioneers. And they recently graduated from Johns Hopkins with a PhD, Sam in 2022, Kiresh in 2023. And my current team here at Santa Cruz, as of 2024, is shown here. And here are the pioneers that are going to be answering the question, what is the mechanism? So I'll stop there and take any questions.
Okay, thank you so much, Carol.
And we can leave those up because I think there might be some other ones that I have that might answer people's questions that are
awesome.
hidden.
Okay, excellent. Yeah, as people are gathering their thoughts, I'm going to kick off with a very basic question. So TTA-GGG, is that the same on any species, or what's the significance of that?
So it tends to be very GT rich, but it varies across species. And so in yeast, it's a more irregular sequence, TG, 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 GG. That's kind of irregular. There's TTG4. So different organisms have slightly different sequences. But in general, they play the same biological phenomena.
Interesting. Okay, questions? Todd?
Are those sequences underrepresented in the regular genome? Wait for the mic.
Oh, the mic.
Oh, yes, we have a mic. Sorry.
Yep. Is this on?
Go ahead.
Okay. If you look at species specific, are the telomere sequences underrepresented in the normal genome?
There are a lot of interstitial telomere repeats, and that's something that confounds our analysis if we're looking throughout the whole genome. That's why we need to look only at the very end. So there are just throughout the chromosome some tracts of these TTA-GGGs. There's also some cases where you had ancestral chromosomes, like in the ape. There's one chromosome that now in the ape it's 2 chromosomes, in human it's 1 chromosome, and at that junction you have head-to-head of those repeats.
Wow.
So there are some internal sequences, and so our bioinformatics needs to be careful to look only at the ends.
Cool.
We'll take a question from online if Ming has any.
Hello? Yes, so we have a question here. Have you looked at base modifications such as methylation on telomeres in your nanopore data? And if so, have you seen anything surprising that you couldn't pick up previously with traditional methods?
So we're just starting to look at base methylation. Obviously, the question comes from somebody that knows a lot about Nanopore. We have a collaborator from Johns Hopkins, Winston Timp, who's an expert in looking at methylation, and he's just now looking at these sequences that we derived. So it's a very interesting question.
Phil?
Yeah, I was just curious about your knockout mice. It seems like why don't you have defects in the first generation? Just because the hematopoietic stem cells are dividing, many, many times. Wouldn't you expect a defect there?
So we didn't find any defect in the first generation, and one of the differences is that laboratory strains of mice have somehow artificially longer telomeres than wild-derived mice. So it gave us an experimental way to look and see only when the telomeres got to be very short was there an effect. We subsequently went back and did that exact experiment again over 6 generations with mice that were derived from the wild. And then you do see an effect in G1 and G2. And those telomeres are more like human telomeres. So yeah, you're exactly right.
Question here at the front.
Yeah. You showed early on that you had a relatively good 3D structure, including the RNA structure in the telomerase enzyme itself. Are there any clues in that indicate to you that it's that enzyme that's able to tell the difference between the different chromosomes? That's what fascinates me, is that both the difference in parental and the difference in the individual chromosomes, there's some way that biochemistry knows which chromosome is which.
Exactly, that's the question. How does it know to elongate one more than the other? We don't think that there's a difference in the telomerase per se. There's been a lot of advances in the cryo-EM and the structure of the telomerase enzyme, and there's just one telomerase enzyme. So we think what it is the particular proteins that bring telomerase to the telomere. Before, we didn't know that there might be different proteins on different ends, right? You thought that all the ends were going to be equally accessible by telomerase. Now our idea is that there's some other component that must be recruiting telomerase more to some chromosomes than to others. But we don't know what that is yet. We're just learning that.
Mayan, would you like to go again?
Yeah. So we have a question here. What happens to telomeres in non-dividing cells, such as neurons?
Yeah, so we did that experiment early on when we were looking at the telomere shortening with replication. You can look in specific tissues, such as neurons, and in those cells that aren't dividing, there is no change in telomere length over time, but there are some neurons, like in the subventricular zone, where there is some division, and then you find telomere shortening. You can also do this in culture, where you can take cells that are actively growing in culture, or you can have them become confluent, so that they age with time, but they're not dividing. And then the telomeres don't shorten if they're not dividing. So it's a cell division phenomenon, not an age phenomenon.
And is there a change in telomerase quantities itself? Or that, like, what do you think is the change in why cell division is driving these changes?
Well, the cell division drives the telomere shortening. The shortening occurs through division, and then telomerase has to, right? So if you don't have the shortening, then you're not going to have the lengthening.
And as we get older, does telomerase change at all, do you think, or it's not?
I don't think we yet have evidence for telomerase changing with age.
Question.
So getting to the cancer question, then this is a naive question, I'm sure, but cancer is said to be a disease of age, and the older you are, the likelier it is to develop, although I know there are a lot of younger people developing it now. And so that doesn't seem to line up with the telomeres being shorter the older you get.
So the telomeres will shorten with age, and then if there's a change that causes those cells to divide more times, that tends to activate telomerase. So the cells will divide, and so they start to get shorter, but now they are being driven, and only those cells that have telomerase can continue to divide. The cells that don't have telomerase will just die like the stem cells die. So you basically select for cells that now have telomerase, and then the cancer can grow. It's kind of counterintuitive that cancer cells have short telomeres, but they're stable because they have telomerase. But we do know that if you have, in these families, I showed you families where they have not enough telomerase, and so they get these degenerative diseases in families that are born, the individuals are born with long telomeres, they're predisposed to melanoma and glioma. So they have a higher predisposition to get these diseases if you have the ability of those cells to now divide more times.
Okay, that is mind-boggling.
Yeah, so that's why it is on both ends. The longer telomeres allow the cancer cells to grow because they are longer telomeres. They don't reach that threshold.
We'll take an online question.
Do we know how telomerase gene expression is regulated?
There's a lot of research of telomerase gene expression. People have looked at a lot of the regulation of the promoter. One of the interesting things is that people are very interested in these days in making what are called induced pluripotent stem cells, or iPS cells. And one of the factors that goes into this, called the Yamanaka factors, you got the Nobel Prize for discovering these factors. One of those factors is a gene called MYC, which specifically activates telomerase. So anytime people are making these induced pluripotent stem cells, they are activating telomerase.
What was your question?
My question was along the same line. I was wondering how total amount of telomerase in cell is regulated, and is there any correlation between length of telomeres and the abundance of the enzyme itself?
Yeah, I don't think we know the answer to that question because most of what we know, specifically in human cells, comes from these assays that we've done, which is flowfish, which is the assay that's used in the clinic because it's reproducible. And that uses blood cells. So we have a lot of information about what goes on in blood cells. What happens in other tissues? We haven't had a good way to measure the length in other tissues. Perhaps this nanopore sequencing can allow us to do that, where you can look at age in different tissues rather than just looking in blood. So it's an interesting question, and I think this opens up possibilities.
Any online question, Maya?
Yeah, so question here is in terms of the research with the short telomere family. Do you know how the disease itself is transmitted, since it seems like from the family tree, some progeny was affected, though some were not?
Yeah, those people that are interested in human genetics, it sounds like the question asker is interested in human genetics. If you look at those pedigrees, it follows what's called autosomal dominant inheritance. And we were able to show with our knockout mice that that is because telomerase is haploinsufficient. Okay, so you can be autosomal dominant, which means only one of the two copies of the gene that you have has to be mutant, and you still see the phenotype. So there's something called a dominant effect, where if you have one mutant and one wild type, sometimes the mutant will take out the wild type. The alternative is you have one mutant and one wild type, and it's just not enough to have only one wild type. And we were able to show with a mouse that that is the case. And so the families show that same inheritance, that if you have a mutation in the gene, even though you have another good copy of the gene, you still see the effect. So it's an autosomal dominant inheritance pattern.
Question here at the front?
Is the average telomere length at average telomere length at any given age correlated with lifespan?
So we can tell you for sure in the mice that it's not, because we can look at lifespan of mice and telomere length, and there is no correlation. In humans, it's been clearly shown that there are multiple different inputs into total lifespan. So the telomere lengths are not directly correlated with total lifespan. Let's imagine that you have 20 different things that contribute to lifespan. If you were to change just one of them, the telomere length, you're not going to change the lifespan because you have 20 things that are determining the lifespan. So it clearly plays a role in specific age-related degenerative diseases, right? So in these diseases that I told you about, the bone marrow failure, pulmonary fibrosis, the GI disease, so there are certain diseases where the short telomeres play a role. There are other diseases of aging that it doesn't play a role, but maybe some of these other pathways play a role there. So there are multiple different inputs.
We'll take an online question.
Are there any evidence of structural defects in telomerase that contribute to its regulatory function?
Yes, there are. So it's known that the telomerase has to interact with specific proteins that are bound to the telomere to be able to elongate the telomere. And so there are specific changes in the telomerase protein where it can no longer make that interaction, and so it will no longer interact with the telomere. So there's a very rich literature on the structure and function of telomerase.
Any other questions? Todd?
So you've been talking about mutations in the telomerase protein, but what about the RNA? Are those autosomal dominant, or is there more ability to deal with mutations in the telomerase RNA?
The mutations in the telomerase RNA and the mutations in the protein component have the very similar phenotype. That's seen in the patients. Of course, the protein is a larger target, so you're more likely to hit that with a mutation. The RNA is just 400 base pairs long. But we tested that directly in the mouse because there was a question. We'd made a telomerase RNA knockout mouse, And so we went back and we made the protein component knockout mouse and bred it for 6 generations and did blinded studies with the pathology, and it was exactly the same phenotype. There was a question about whether there might be a different phenotype, and it matters for the patients whether you have a mutation in one or the other. So that's why we spent $400,000 doing that experiment.
Very good.
Question here?
So where is this, what's the future of this? And if you see evidence of this shortening happening in a person, say, is there going to be an intervention, or where is this going?
Yeah, people have been talking about various kind of interventions. Like we were interested in the role of telomerase in cancer. I wrote a review article in 1990 about the role of telomerase in cancer, and maybe you could inhibit cancer cells by inhibiting telomerase. That's not there yet. But now that we know that there are certain people that are susceptible to short telomeres, it's got to go to individualized medicine. So you would need to know, if you had a way to inhibit telomerase, you would need to know what is the general telomere length of this person, and would you be doing them harm by inhibiting telomerase? Likewise, on the other side, would you want to increase telomere length? Is there already long telomeres? Would you be susceptible to cancer? So it's going to come down to individualized treatment. But now that we know it's not just one equilibrium, we have to look at every chromosome. Is there one chromosome or 2 chromosomes or 3 that are the sentinel chromosomes, that ones that get shorter? Or is the shortening on chromosome 10 the same effect of the shortening of chromosome 7? And we don't know that at all. What we're really interested in looking at is what is the mechanism that allows chromosome 1p to be different than chromosome 2p. Because once you understand that mechanism, then you can start to think about ways that you could do these disease treatments.
We'll take an online question.
Yeah, are there any extrinsic factor that govern telomere length on top of intrinsic ones?
There is a large literature suggesting that there may be extrinsic factors, although the methods that they use is typically what's called quantitative PCR. I didn't present that as one of the methods. And it has a variation of about 15 to 20%. And so I haven't yet seen evidence with a method where you can show changes in telomere length over time that says that there are extrinsic factors. I'm not saying that there are not, but I have not yet seen evidence that there's any thing that one can do to change one's telomere length.
I think there's a question over in the back. Yeah.
So do they have telomere structure on the circular DNAs?
Oh, circular DNA?
Yes.
No, so bacterial chromosomes
Yeah,
don't need telomeres?
some circular DNA is in human mitochondria or something.
Yeah, the human mitochondria will be like bacterial chromosomes in that they originally came from bacteria. So they have solved the problem of replicating the end of the chromosome by having a circular chromosome. There are some examples of actually mitochondrial DNA that's linear, and they have a different way of solving the problem of the telomeres. It's not a telomerase-mediated mechanism. So there are multiple ways that this problem has been solved. It turns out that all eukaryotic organisms, not all, most, Drosophila, don't do it. Most eukaryotic organisms do telomerase. There are a few exceptions.
You showed a really interesting graph of the difference between the parental versus the maternal. What do you think is driving like a sex difference in a chromosome?
Yeah, I mean, that's very new data. But it was interesting. So I pointed out that the 1p maternal and 1p paternal were very different. But if you look at other chromosomes, it goes the other way. So it's not always the paternal one. So we're very interested in what are the DNA sequences are there, and maybe there are proteins that are binding to those DNA sequences that are driving that. And that's what we're getting at both in yeast as well as in human cells. Of course, in yeast, you can do it a little bit faster. But there are slightly different questions to ask in the different realms.
We'll take one more question. Anne.
I wonder if you, what's known, if at all, about the mechanism between shortening of some of these, the telomeres and those age-related diseases, like what's actually the molecular mechanism going on causing the
Yep.
age-related disease.
So when the telomeres get short, and the point I was trying to make is that there's a threshold. So it's not like you have a long telomere that's now getting shorter and there are different kinds of biologies. There's really no biology that we can get at until it gets to be critically short. And what we've shown in yeast and many other people have shown in mammalian cells is that that short telomere now doesn't look like a telomere anymore, and it induces a DNA damage response. So it's an ATM mediated through P53, so very classic DNA damage response when the telomeres get to be short, because they're not recognized as protective telomeres anymore.
Well, thank you, Carol, for a fascinating lecture. And thank you, Susan, for moderating an equally fascinating after-lecture discussion. Our next lecture will be on March 13th. It will be concerning computational data, and speaking will be Assistant Professor David Lee. Thank you all for coming tonight, and I hope to see you at the next one.